Hello students, this is Dr. Anjani, Assistant Professor in Biotechnology. Welcome to e-learning sessions in cell biology. Today in this session, we learn about structure and functions of Golgi complex. Golgi complex was discovered by Camillo Golgi during an investigation of the nervous system, and he named it the internal reticular apparatus. Functionally, it is also known as the post office of the cell. It is considered as post office of the cell. Certain important cellular functions such as biosynthesis of polysaccharides and packaging of proteins and production of secretory vesicles and differentiation of cellular membranes occurs in the Golgi complex. The Golgi complex or the Golgi apparatus is located in the cytoplasm of animal and plant cells. Coming to the occurrence, Golgi complex occurs in the eukaryotic cells. It is present in the eukaryotic cells. Exceptions are the prokaryotic cells, including mycoplasma, bacteria and blue-green algae, and eukaryotic cells of certain fungi, sperm cells and cells of mature seed tube cells in case of plants, mature sperm cells and the red blood cells in animals lack this Golgi complex. Coming to the morphology, Golgi apparatus is morphologically very similar in both plant and animal cells. However, it is extremely pleomorphic. In some cell types, it appears compact. Appearance will be differing from one type of the cell to another cell. In some cells, it is compact and limited. In others, it spreads out like the membranous structure all over the cytoplasm. Its shape and form may vary depending on the cell type. Its shape as well as the number and the form it appears vary from one cell to another cell. It appears as a complex array of interconnecting tubules or vesicles or cisternae. There has been much debate concerning the terminology of the Golgi parts or the Golgi apparatus. The smallest unit of the Golgi apparatus is the cisternae. This is a membrane-bound space in which various materials and secretions may accumulate. Numerous cisternae are associated with each other and appear in a stack-like aggregation. A group of these cisternae is called dictyosomes. Group of cisternae is called dictyosome. A group of dictyosomes make up the cell's Golgi apparatus. All dictyosomes of a cell have a common function. The detailed structure of the basic components of the Golgi apparatus are described in the coming slide. So, this is the slide which explains the different forms of the Golgi apparatus involved in packaging of the biomolecules or it acts as the master or the post office of the cell. It has the flattened sac-like structure. They are called as the cisternae. It has the flattened sac-like structure. They are called the cisternae, and it has the highly reticulated structure. They are called as the tubules, and it is also having the vesicle-like structure. So these vesicles will be having the packed material inside it. We will take up this cisternae first. So this cisternae of the Golgi apparatus is about 1 micrometer in diameter, and it's a flattened plate-like structure or saucer-like closed compartments which are held in parallel bundles or stacks, one above the other. In each stack, cisternae are separated by space of about 20 to 30 millimeter, 20 to 30 nanometer, which may contain rod-like elements or the fibers. Each stack of cisternae forms a dictyosome, which may contain 5 to 6 Golgi cisternae in animal cells, or it may be 20 or more cisternae in case of plant cell. So the number of cisternae from one cell to another cell will be varying. Each cisternae is bounded by smooth unit membrane having a lumen varying in width about 500 to 1000 nanometer. So the diameter, the width of the cisternae will be varying. The margins of each cisternae are gently curved. The margins of each cisternae will be gently curved, and the cisternate at the convex end of the dictyosome comprises proximal, or it is forming, or the cis phase, and the cisternate at the concave region will be the region where it releases the packed proteins or the biomolecules to the cytoplasm. And it also has the complex array of associated vesicles, and they are called as the tubules. So even tubules play an important role in packaging of the material. Along with that, it also has the vesicles. So these vesicles will be carrying the important biomolecules that will be packed at one end, and it will be secreted on the other end. So it will be having the two regions, where one end there will be the entry of the biomolecules takes place, on the other end there will be releasing of the particular biomolecule takes place. So it is continuously packaging the materials or the biomolecules which are required by this cell. So that is regarding the structure of the Golgi complex. Next, coming to the functions, it is considered as the post office of the cell, which is involved in the modifying, sorting and packaging of the macromolecules or the biomolecules for cell secretion. So this Golgi complex is involved in the transport of lipids around the cell and the creation of the lysosomes. It is involved in the synthesis of the lysosome, which is considered as the suicidal bag of the cell, which is filled with the lytic enzymes, or the lyases will be formed by the Golgi complex. Proteins are modified by the enzymes in the cisternae by glycosylation and the phosphorylation. So this glycosylation and the phosphorylation will make the further post-translational modification of the proteins, and they will be packed inside the Golgi complex. In the membrane that will be considered later as the lysosomes. For example, the Golgi apparatus adds mannose-6-phosphate labeled to protein and destined for the lysosome. So the proper transportation of the proteins or the enzymes in the particular region inside the cell will be done by this Golgi complex. So that's why it is considered as the post office of the cell. Next, this Golgi complex also involved in the proteoglycans and the carbohydrate synthesis. Golgi complex is not only involved in the packaging, it is also involved in the synthesis of certain biomolecules that includes the proteoglycans and the carbohydrates. This includes the production of the long unbranched polysaccharides, which the Golgi then attaches to the protein, and it results in the formation of the endoplasmic reticulum to form proteoglycans. So these carbohydrates will be synthesized by the Golgi complex, and that play an important role in helping this the endoplasmic reticulum. Golgi it is involved in transportation, packaging and sending the biomolecules to the particular site, and it is also involved in the synthesis of certain biomolecules which play an important role in other cell organelles. Apart from that, in animal cell, Golgi complex is involved in packaging of the biomolecules, and it is also involved in exocytosis. It is involved in exocytosis, and this exocytosis will be very specific. So the particular biomolecule will be reached at the particular site of the cytoplasm. For example, zymogen of the exocrine pancreatic cells are the mucus secretions by the goblet cells in the intestine. So the secretion of the particular compounds by the cells, the secretions of certain hormones like the thyroid hormone, in case of the thyroid thyroxine that will be produced by the thyroid gland will be done packaged, and it will be done by the Golgi complex. So apart from that, it is also involved in the formation of certain cellular cell organelles such as plasma membrane, lysosome, acrosomes of the spermatozoa, and the cortical granules of variety of oocytes. So since it is involved in the packaging and exocytosis, it is involved in the synthesis of the plasma membrane. After it excretes or throws out the packaged material, the remaining membrane will be involved in the synthesis of the plasma membrane. Apart from that, since it packs the hydrolytic enzymes of the lysosome, or it forms the vesicles of the membrane which are filled with the lytic enzyme, they functions as the lysosome. So the lysosomal membrane proteins as well as the lytic enzymes will be bundled, or it will be synthesized, it will be packaged in the Golgi apparatus, and it is involved in the plants. Golgi apparatus performs various functions in the plants. That includes, it is mainly involved in the secretions of the materials of primary and secondary cell walls. So that is involved in the formation of export of glycoproteins, lipids, and other biomolecules in case of the plants. During cytokinesis of the mitosis or the meiosis, vesicles originating from the Golgi apparatus coalesce and they result in the formation of the cell plate. The cell plate formation, or the membrane which is required for the cell plate formation in case of the plant cell division, is derived from the Golgi apparatus. So that is the difference between the plant cell division and the animal cell division. In the plant cell division, the cell plates are formed at the center of the cell in dividing the parental cell into the daughter cell. So that membrane is derived from the cell plates are nothing but the short fragments of the membranes. So that will be formed by the Golgi apparatus. Later they coalesce, they rejoin, at their end will be joining, and they result in the formation of the two daughter cell. So that function will be done by the Golgi apparatus. So this is regarding the functions of the Golgi apparatus. So in this session we learned the structure of the Golgi apparatus and few functions that will be performed by the Golgi apparatus. I hope you understood this session. Thank you.